Hello everyone, welcome back to Microbiome Solutions. Today we are going to discuss about motility testings used for bacterial motility identification. Motility is the ability of the organism to move itself by means of propeller-like flagella, which is unique to bacteria or by special fibrils that produce a gliding form of motility. Bacteria possess flagella and move using the flagella in a thread-like locomotor appendages extending outward from the plasma membrane and cell wall, either single or multiple flagella. So, a motile bacteria, has got a flagella, which is a thread-like structure which helps the bacteria in moving around. The flagellum can be single or multiple. Based on the type of flagella, bacteria has been further classified. Each flagellum has a very rigid helical structure, and actual motility results from the rotation of the flagellum in a manner similar to that of a boat propeller. Hanging drop preparation is one of the easiest methods to observe the motility of the bacterium. This method is carried out by putting a loop full of bacterial suspension on the cover slip and placing it over a cavity slide, a slide with an excavation inside. An advantage of this method is that the live bacteria can be observed. You can either take it from the sample in case of Vibrio cholerae infection. So the Vibrio bacteria will be present in the rice water stool, which is a characteristic clinical feature of the bacterium. In such cases, you can directly take the rice water stool of the patient and do a hanging drop method, or in case other bacterium to be detected for motility, you can take the culture plate and emulsify it with either saline and then observe under microscope. Examination of living organisms is useful to observe the cell activities such as motility. The principle of hanging drop test is microorganisms such as bacteria, because of their smaller size and they have got the refractive index which closely approximate that of water and do not lend themselves readily to microscopic examination in a living unstained stage. And bacteria, they are motile due to the presence of different types of flagella, and depending on the location of flagella, bacteria can be classified. Spirochetes are a group of bacteria or organisms that are examples of being motile but without the presence of an external flagella. Spirochetes has got an internal flagella which helps in the locomotion of the organism. The requirements for this particular motility testing, you need to have a compound light microscope, normal saline, inoculating loop, cavity slide or concavity slide, cover slip, log phase broth culture of bacteria. If broth culture is not available, you can take the culture of the bacteria from the nutrient agar plate and emulsify the culture in normal saline and use it for hanging drop method. Procedure: you need to take a clean, grease-free cavity slide and also a cover slip. Once you've taken the cover slip, you have to apply paraffin or Vaseline to the four corners of cover slip and place a drop of broth culture or the culture emulsified in normal saline on the center of the cover slip with the help of an inoculating loop. And place the cavity slide, keeping the cavity down on the cover slip so that the drop is placed in the center. Invert the cover slip and observe under microscope. First, focus under 10x to locate the edge of the drop. So remember, you have to focus the edge of the particular drop where the bacteria tends to move towards the edge of the drop where they get enough oxygen for living. And shift the focus to high power 40x and observe for motility. So this is the slide which is showing hanging drop method. This is a cavity slide. You can see a center cavity in the slide. And then this is a cover slip. On the cover slip, 4 corners of the cover slip, you will be applying Vaseline or paraffin. And you can see this picture you have applied. You can see, if you clearly make out, you can see the 4 corners of the cover slip has been applied with paraffin or Vaseline, and there is a central inoculum of the bacteria, broth culture or the normal saline culture mixture. And then this cavity slide is taken with the cavity down and it is placed on the cover slip with the cavity center and the drop at the center. So this cavity slide is kept on the cover slip and then it is inverted immediately. Since it has got this paraffin or Vaseline, this cover slip will stick onto this cavity slide, and if you invert it properly, invert it drastically but not vigorously, you will be able to see this drop hanging from the cover slip to the cavity. That is what it is called as hanging drop technique. So this drop will be focused under 10x and 40x simultaneously for identifying the motility of the particular organism. The quality control used for this test is a motile culture of Pseudomonas aeruginosa for appropriate details such as motility, natural sizes and shapes of the bacteria. So it is important to differentiate about active motility from Brownian movement. There is something called as Brownian movement or false motility or pseudo motility. So it is very important to differentiate the active motility of the bacterium from Brownian movement. Brownian movement is not a true motility. Instead, it is exhibited due to the movement of organism as a result of their collision with water molecules, or the bombardment of water molecules also can lead to Brownian movement. This movement is usually seen around the axis of bacteria. And next technique which can be done in the lab for motility is Mannitol motility testing. In this testing medium, you can also find out the fermentation of mannitol agar by the bacteria. Motility by bacterium is demonstrated in a semi-solid agar medium. That is what the principle of MMR or mannitol motility medium. This medium mainly used for the purpose which is a combination of mannitol sugar and motility. As it is a semi-solid agar, this has got a very soft consistency which allow motile bacteria to migrate readily through them, causing cloudiness of the medium. The inoculum is stabbed into the center of a semi-solid agar deep using a sterile inoculating needle. So this medium will be held on a tube and the medium will be stabbed into the center of a semi-solid agar. And bacterial motility will be evident by a diffuse zone of growth which is extending out from the line of inoculation. And some organisms grow throughout the entire medium, whereas others show small areas or nodules that grow out from the line of inoculation. And the non-motile bacteria will only grow in the soft agar tube and only the area where they are inoculated. So you will not be able to see any diffused growth if the bacteria is non-motile. Only in the stab line the bacteria will be grow. So the principle of Mannitol motility test, this medium is designed to differentiate bacteria on the basis of their motility and also their ability to ferment mannitol. Fermentation of mannitol produces acidity in the medium, and phenol red is used as a pH indicator which detects the acidity in the medium by exhibiting a visible color change from red to yellow. So if the organism is able to ferment the sugar mannitol, the color of the medium will change from red to yellow. The procedure of inoculating mannitol motility medium is using a sterile straight wire, touch a colony of a young culture, preferably 18 to 24 hours old culture, which is growing on an agar medium, nutrient agar medium usually. Single stab down the center of the tube to about half the depth of the medium and incubate the mannitol motility tube at 35 to 37 degrees Celsius and examine next day. So the interpretation would be motile organism will spread out into the medium from the site of inoculation with a diffuse zone. You can see in this last picture the motile organism is spreading out with a diffuse zone, whereas the non-motile organism remains at the site of inoculation. So this is a tube which is showing a non-motile organism, and this is a tube which is showing a motile organism. The fermentation of mannitol is not shown in this picture. So this is an uninoculated tube with a red medium, and this is how you put your stab line. And if it is a non-motile organism, the organism will remain at the site of inoculation, and as it is a motile organism, it has diffused out from the stab line. The quality control used for mannitol motility testing is positive Escherichia coli ATCC 25922 and negative Staphylococcus aureus ATCC 25923. This is about today's class. If you have any queries, please post it in the comment section. Please like, share and subscribe to our channel. Thank you.